We've saved the best for last, is what we say when we make one of our colleagues wait till the end to speak, but this is true today. One of the greatest pleasures of my career is to be working side by side and to host this meeting and to run the cutaneous program here at Angeles Clinic, Cedars-Sinai with Dr. Mark Sayre. And I won't say more because he'll show it to you. He's an amazing physician, surgeon, and speaker.
All right. Thanks so much. I've enjoyed the sessions and give the audience a sense of relief that try not to include too much more data out in it. Lots of information, lots of good information. But I'll try and go back to the beginning in some ways and bring you up to speed a little bit in terms of where we stand with cellular therapies, T-cell therapies. And so in terms of what I'll cover, what is a T-cell? What are we even talking about? What does it do? How does it work? Why are we interested in it? How do we come to use T-cells as a treatment? Why did it take so long to get here? And then how are we improving this therapy as we move into the future? So, basically we know and have known for a long time that the immune system has the ability to detect cancer, to recognize it as abnormal and even attack it. So this is just a slide of a biopsy under the microscope of a melanoma, just a melanoma in the skin. And melanoma cells are the big cells at the top. And all of those little dark blue cells at the bottom are lymphocytes, many of which are T cells. And you can see a few of them are making their way up into the tumor. And they're not there by accident. And we actually didn't do anything to make them go there. They just went there on their own. On their own, they were able to recognize that there was something wrong with these melanoma cells, and they are trying to deal with them. And so this has been known for a long time that this happens. And these immunotherapies that you're hearing about are ways in which this ability has been taken advantage of as a form of treatment. And so this goes way back. So this William Bradford Coley, he was a surgeon in New York at the end of the 19th century. And he noted that occasionally a tumor might get infected as a result of growing too much and then would spontaneously regress, go away. So he literally cooked up mixtures of bacterial toxins from various cultures and injected those into tumors. He knew that he had gotten the right dose. The patient developed a fever to 102 or 103, and actually saw quite a number of regressions of tumors that he injected, but it wasn't very reproducible. And at the time, it was a one-shop sort of operation, and was really able to spread out to other locations in a meaningful way. And when other cancer therapies came along, it sort of fell by the wayside. But it has been maintained, this sort of idea has been maintained specifically in melanoma over the years, really since the 70s and 80s. And my mentor, Don Morton, developed the concept of injecting something called BCG into melanomas. And we still occasionally do this, but this is similar to the TVAC that you've heard about, the RP1 that you've heard about, where we inject something into the tumor to cause something to happen there, the hopes of making other tumors go away with that injection. So this is just a case of a woman that we treated years ago who had what are called in-transit metastases. These are metastases of melanoma in the skin, and you can see she has hundreds of them on her leg, backside there, and so no way to operate on this, no way to do regional chemotherapy for it. This was before the days of Yergoi and Keytruda and such. And so we injected a proportion of these tumors with BCG, and you can see there was inflammation that developed at the sites of the tumors that we injected. And those tumors that we injected went away, but the rest of the tumors went away too. And so these spots that you see that are left there now are just pigment tattoos, basically, and all the melanoma was gone. So this really, even before we knew that much about the immune system, proved the concept that we could harness the immune system to kill tumors. And so what's happened is there's been an explosion of effective therapies in melanoma. You could see through the 70s and 80s and into the 2000s, we had almost nothing to offer, very, very little offer. And then there have been this raft of treatments that have come along. All the ones that are in the blue boxes are immune therapies, and they are all dependent on T cells to work. How does all this operate? So if you take a cell, any cell, and look at how it works, how it functions, there's DNA in the nucleus, and that then produces mRNA, same things that are made in these vaccines, but the body does it on its own. The mRNA is then passed through a ribosome and are converted into proteins. And one of the ways that the body works to sort of maintain itself, these proteins are broken down into something called a proteasome, the peptides, and those peptide fragments are then loaded into something called MHC or HLA. And so when you have this tissue typing done, when you look to see if you're eligible for some of these bispecific antibodies, those sorts of things, this is the specificity that's being looked at, these immune cells. And these are really the context in which these peptides are presented. This is a way for the cell to say what's going on inside the cell. And the importance of all of this is that is how T cells determine whether a cell is a good cell or a bad cell. So the T cells then come into play. They come in, interact here, and take a look at what peptides are being presented. So if there are mutations in the proteins, mutations in the DNA that lead to abnormal proteins, neoantigens, those get loaded in here. The T cells recognize that it's not a normal cell and know to kill it. And the T cell's job is to kill those cells. And so the way a lot of these therapies work is we manipulate this interaction in a way that enhances the ability of those T cells to kill the melanoma cells. There are those T cells again, you saw. So in terms of immunotherapy, you have melanoma cells, T cells go and find them, hopefully kill them. That the dying cells potentially that release antigens that can be picked up by antigen-presenting cells, another type of immune cell. They can then mature into these dendritic cells that are very effective at stimulating and multiplying that immune response. And we can impact that. That can be impacted by helper cells, it can be blunted by regulatory cells. And we now have these drugs that we can use that work through the system. So one of the drugs that you're familiar with is Yervoy or ipilimumab. And that probably works at this interaction between the antigen-presenting cell, the cell that's supposed to stimulate the T cell to go off and do what it's supposed to do, educate it basically. And there's an interaction between the antigen-presenting cell and the T cell with CD28 that activates that T cell. It then goes off to find the melanoma cells that kill them. And then eventually it circulates back around and may interact again with the antigen-presenting cell. But now it has something called CTLA-4 on its surface instead. And that interaction actually shuts that T cell off. It's a way the immune system normally turns itself off when an infection is clear, that sort of thing. But this can be used in the setting of tumors for the tumors to escape. And so Yervoy is something that blocks that CTLA-4, keeps that happening, keeps those T cells turned on. That's sort of like a handbrake in a car. The other kind of brake in this system happens actually directly between the T cell and the melanoma cell. So more like foot pedal brake. And here, those cells come into contact with this interaction. The T cell potentially could kill that tumor cell, recognize it as normal, but it may have this PD-1 on its surface that can interact potentially with PD-L1 on the surface of the tumor cell. And that interaction shuts that T cell off as well. And so the Keytrudas, the Optivos, they keep that from happening and allow those cells to continue on to kill those melanoma cells. And so we have learned in this cycle of the tumor cells, the immune system, how to manipulate the interactions. And so we can interact at the CTLA-4, PD-1, we've heard about LAG-3, but it's not that simple even. There are ways of killing tumor cells to release more antigen, produce more immune response. You can increase the maturation of dendritic cells, various things, and presentation of antigens. You can affect the other regulators of the immune system. You can apply various cytokines and binding proteins. And anyway, it gets a little complicated. And for the simple-minded surgeon, it may be, above my level. So we think about just using T cells, a fairly simple concept, that just sending them in, sort of like a cruise missile, fully packaged killing machines. And how can we do that? And that's this adoptive immunotherapy, T cell based immunotherapy. And so this is a therapy that has been was pioneered by a guy named Steve Rosenberg, who's led the surgery branch at the NCI for many years, and just sort of emphasizing the incredible contributions that the NCI, the National Institutes of Health have made to cancer research over the years. And so the idea that Dr. Rosenberg had was that we know that these lymphocytes can infiltrate tumors in the body. We can surgically take those tumors out. And if you look at the tumors outside of the body, you can find those T cells that are in there again. But you can take some of that tumor tissue and actually put it into culture in the lab in conditions that favor the growth of the immune cells. And those immune cells will grow up in those culture wells, kill the tumor that's in there. And then you can take those T cells, actually test them outside of the body and see if they kill melanoma cells outside of the body. And you could also expand them through a culture conditions called rapid expansion protocol to increase the numbers to many billions of cells. And those cells then can be given back through an IV to patients with melanoma with the hopes that those T cells will be strengthened in numbers and ability so that they can then kill the tumor cells that are in the patient. And that clearly works. Known it has worked for some time. These are just case examples that came from the NCI of tumors that shrink, go away, often in rapid fashion after its infusion of these cells. But this is a fairly complicated treatment, as you can imagine. And for many years, it was only done at places like the National Cancer Institute or MD Anderson, places you need to Google Earth to be able to actually see the whole place. And so it wasn't felt that it was going to be practical to be used outside of those sorts of places. Dr. Amita and I were crazy enough to start our own TIL protocol years ago and added to the cellular growth lab. And there's a cellular product that we made that we gave to a patient. But it was a pretty heavy lift and wasn't going to be something that would be mainstream. Now it is mainstream. And that is due to collaboration with commercial partners that have enabled the production of these cells in a practical way. So the first cells to the finish line for this were CAR T cells used in lymphomas, leukemias, and there are many companies that were involved in developing those CAR T cells, CAR NK cells, and then TIL, the main focus here, these tumor-infiltrating lymphocytes that have grown, and a bunch of companies that are working on that. The ones that were first to the finish line in terms of approval of FDA approval for product were Iovance and Adaptimmune. Both have FDA approvals now for their products, and then they're available now outside of a clinical trial. The trial that led to the approval of that TIL treatment from Iovance was this one. You see Dr. Mead's name there as second in the list. And the way the process works from the patient's perspective, there's a tumor that's taken out. It is cultured through this multi-step process in the lab. The culture process itself takes about 3 weeks, a little over 3 weeks. From the patient's perspective, they have the tumor removed, surgically removed. And then this period where they're waiting and that process goes on. It's actually probably more like 5 or 6 weeks. Typically, it takes between the time when the tumor is taken out and the cells are given back, because there are quality control things that need to happen and other things that delay the treatment itself. When the patients get the cells, they have been recently treated with lymphodepletion. It's chemotherapy that's designed to get rid of the lymphocytes that they have in circulation already and essentially create space for the lymphocytes that are about to be infused. And then they get this interleukin-2, that cytokine that you've heard about, that's a lymphocyte growth factor that supports those cells after they've been infused. And this is that waterfall plot. It's maybe the only plot that I have for you guys that shows, these downward bars, and that represents shrinkage of the tumor. And you can see that happen in the vast majority of the patients. There was some shrinkage of their tumor. The patients that experienced responses, those responses, most of the time were durable. So lasted for over 2 years in this level of follow-up. So it seemed like this one-time, once and done sort of treatment did have a lasting benefit for the patients that responded well. And so it had approval last year. There are a lot of challenges with this therapy, though. It is not for everyone. There is the delay in terms of timing between when the tumor is harvested and when the patient actually gets the treatment. You need to have surgery to get the tumor out. You need to balance the risk of procedure for removing the tumor with being able to get a sufficient sample to be able to create the therapy. There's toxicity associated with chemotherapy they get preparation, and there's toxicity that's associated with that IL-2 they get to support those cells afterward. And then it doesn't work for everyone. Even when we get cells for people, it doesn't cause the tumors to go away for all those patients for a variety of reasons. And so those are the areas, all of these areas are areas that there's work being done to try to improve the therapy to make it available and effective for people. Just an example of a patient that we treated in terms of the surgical planning for this and selecting the appropriate tumor. The patient had a lot of different previous therapies and was now going to get TIL, and a tumor, very small tumor in the liver, a very big tumor in the right adrenal gland, and then another small tumor just in the soft tissue. So for this setting, the small soft tissue lesion was clearly the right one to get to without causing a lot of toxicity from the surgery itself, but providing a sufficient sample to allow it. So how are we altering these regimens to try to improve the therapy? You can step up the timing, moving it earlier, so patients have more time to get there, potentially reduce the intensity of some of these conditioning regimens, selecting better lymphocytes from the tumor to potentially use, combining these therapies with the other effective interventions that we have, one blockade, other novel interventions, and then delivering tumors, the cells in different ways, and then modifying the TIL themselves to try to make them more effective. I'll show you some examples of how that may work. One is with altering the T cell receptor, the thing that makes the T cell able to identify a very, very specific target, and that's a T cell receptor. And so there are a couple of ways of doing that. One is by taking out peripheral blood lymphocytes, and those lymphocytes then can be altered. You can introduce genes into those cells that change from one T cell receptor to another one that we know is very specific for melanoma target. So these are transgenic T cell receptors. The other way is with these CAR T cells, chimeric antigen receptor T cells, where a gene is introduced into these cells, makes them put on their surface what is essentially like an antibody, but it is linked to the same T cell signaling that would be in a normal T cell receptor. And so then these modified things can come up against either the peptide in this MHC, just like the normal T cell kind of interaction, or by recognizing cell surface protein through this antibody type molecule. And these different approaches have pros and cons to them about what the targets can be, those sorts of things. And so both approaches may have value. The other thing that can be done is altering the function of these T cells. So this is just the standard TIL, that is the TIL that have been selected because we already know they make their way into tumors. They have the right T cell receptors to do that already. But instead of relying on exogenous interleukin-2, that cytokine that has all the toxicity to bind to IL-2 receptors, these cells, these are the OBX115 cells that Dr. Memi mentioned, have a gene introduced so that they have membrane-bound IL-15 and other T cell stimulating cytokine on their surface. And then they can be given this small molecule drug that activates that, essentially an on switch for that. And the cells stimulate themselves. You don't have to give the IL-2. So this may be a way of avoiding that toxicity. There are lots of potential immunologic hurdles to get over with immunosuppressive environments and antigen escape and those sorts of things. And there are ways of modifying these T-cell receptors, either chimeric antigen receptors or T-cell receptors, to try and overcome those difficulties. And then a separate type of cell that can be used as a cellular therapy are NK cells. These are natural killer cells that may have an inherent ability to recognize and contain cancer cells. So overall, it's clear now to us that T cells are critical components of immunotherapies, these adoptive immunotherapies, but also the checkpoint immunotherapies you heard about. You need T cells to be working for any of those to work. There are still challenges in applying TIL therapy to patients more broadly that we are working with ongoing research to try to overcome, to try and make these cell therapies available to as many patients as possible. And this, as you can imagine, is an enormous team effort. So the TIL therapy is very complicated and requires a bunch of people with a bunch of different types of expertise. This is the Angeles Clinic group with nurses and medical assistants and pharmacists and many others who make all this come together to work. So appreciate the opportunity to participate in the symposium again.